So welcome back to TMS, everyone. It's a real pleasure this week to introduce Elisa Mouton, who is both a PhD student at the Cancer Research UK Cambridge Institute, as well as a very good friend. So Elisa is from Marseille in France originally, and she did her undergraduate in life sciences at the University of Aix in Marseille in 2017, and then spent the last year of that also in the UK as a part of the Erasmus programme. She did her master's degree in cancer biology from the University of Montpellier and graduated in 2018. And during that time, developed an interest for the study of stem cell biology and cancer. So since then, she started her PhD in the Winton Lab at the CRUK in 2019, and is now studying the impact of chronic inflammation on stem cell dynamics, and specifically its role in increasing cancer incidence in patients suffering from inflammatory bowel disease. So it's great to have you with us, and I will hand over to you for your talk today.
Thank you very much, Anna. So yeah, I'm very happy to be with you today to talk about the subject of my PhD project. So today I'm going to be talking about clonal expansion in inflammatory bowel disease and its role. So when a tissue has been injured, and that can include, for example, a cut on your hand, but also the degradation of the tissue by bacteria, an inflammatory response is necessary to clear the pathogen and to repair the tissue. So as you can see on this scheme here, the immune cells travel to the site of injury to kill the pathogen. And once the pathogen has been cleared, stromal cells, such as fibroblasts, secrete molecules like fibrin, part of the extracellular matrix, to form a clot or a scar. Now, in the case of acute inflammation, the extra immune and stromal cells are then removed and the normal function of the tissue is reestablished. However, when the injury is sustained or repeated, that leads to an excessive immune response and the excessive production of quantities of molecules such as fibrin. This actually leads to more inflammatory cells being recruited to the site of injury, contributing to it. So this is the vicious cycle of chronic inflammation in which the tissue is not able to repair properly and doesn't recover its normal function. Chronic inflammatory illnesses can affect many different organs, and they are very painful, debilitating diseases with comorbidities that can include an increased risk of developing cancer in the organ concerned. And these types of cancers are called chronic inflammation-associated cancers, or CIACs. They are highly lethal with a 5-year survival rate of about 15%. And they often affect young populations and are diagnosed at advanced stages, which means that they have a poor prognosis. And unfortunately, there is also a lack of effective therapies for these types of cancers that present differently from the ones that are not associated with chronic inflammation. So on this drawing, you can see 4 types of CIACs, and these are the ones that are being investigated by the Storming Cancer Team, of which my lab in Cambridge is part of. So this is an international collaboration, and the goal is to study different types of epithelial CIACs, and we also included the lung as an outside control. So in this case, chronic obstructive pulmonary disease is mostly caused by cigarette smoke, and it increases the risk of developing lung cancer. Chronic inflammation of the stomach or gastritis has many possible causes, such as bacterial infection, the regular use of painkillers, and also excessive alcohol consumption, for example. And this is linked with gastric or stomach cancer. Esophageal cancer can arise from Barrett's esophagus. That's a chronic inflammatory illness caused by gastric reflux. And finally, chronic inflammation of the colon, or inflammatory bowel disease, is the pathology that I am studying during my PhD, and the one that I will talk about today. So inflammatory bowel disease, or IBD, is characterized by chronic inflammation of the gut, and it primarily includes Crohn's disease, which can affect any gut segment, the small intestine or the colon, and also ulcerative colitis, which is limited to the colon. This is a pathology that is more prevalent in rich and industrialized countries, but it is actually on the rise in newly industrialized countries like China. And the exact cause for the development of IBD is currently unknown. A possible cause would be an immune system malfunction. But what we do know is that diet and stress can contribute to the worsening of the disease. So both Crohn's disease and ulcerative colitis are commonly present with abdominal pain and diarrhea, and they are associated with an increased risk of developing complications such as abscesses formation and perforation of the guts, but also, as I mentioned earlier, a form of colorectal cancer that is called colitis-associated cancer. The risk of developing colitis-associated cancer is increasing with the duration and severity of inflammation. And the current standard for the diagnosis is through the finding of high-grade dysplasia in the tissue. This describes histologically abnormal but non-cancerous cells. And you can see an example of this in these images. These are tissue stained with hematoxylin and eosin. And as you go into dysplasia, you can see that the colonic crypts are losing their polarization and their architecture. And the lesions that we observe in colitis-associated cancer are flat, very much unlike the classical colorectal adenoma, of which you have an example here. And this makes them not easily detectable. So for this reason, patients suffering from inflammatory bowel disease have to undergo lifelong surveillance with endoscopy, colonoscopy, sometimes chromoendoscopy. You can see an example here with a blue dye marking dysplasia. And they also have to suffer through recurrent biopsies. So this really highlights the need that we have to find better diagnostic tools that are less invasive. And even though the sequence from active inflammation to dysplasia and finally cancer is accepted, the mechanisms through which the cells can advance from one step to another are not well understood, and it could actually help develop better diagnostic tools. So if we now zoom in on the gastrointestinal tract, you can see it is lined by a single layer of epithelial cells called the intestinal epithelium, and it invaginates in crypts in the colon. You can see that in the picture here. These are mouse colonic crypts taken by electron microscopy. At the base of this crypt, you can find intestinal stem cells, and these are a fascinating cell type because they are capable of self-renewal, but also differentiation in all of the other intestinal cell types. So they really fuel the intestinal epithelium and they allow its constant renewal, which you can imagine is very important in a context such as chronic inflammation, where cells are constantly damaged and need to be ejected out of the tissue. So these cells, due to their long lifespan, can also accumulate mutation. And this is why they are thought to be the cells of origin of intestinal cancer. And importantly, they depend on signals coming from their environment or their niche for their survival and for their function. So in the colon, the niche includes stromal cells such as fibroblasts, endothelial cells that form blood vessels, but also the surrounding extracellular matrix, which is made of molecules such as collagen or fibronectin. Unsurprisingly, chronic inflammation is profoundly remodeling the affected tissues, and that modifies the signals that are being shared between the epithelium, the stroma, and the extracellular matrix. And this is particularly relevant as for the extracellular matrix, for example, it has long been thought to be simply a supporting matrix for the cells, but recently it has been explored more in depth for roles such as regulating the bioavailability of growth factors, but also allowing the migration and the differentiation of cell populations through the modulation of its stiffness and its elasticity. So in this context, the Storming Cancer team is looking at all of the ways disruption of signalings in these different compartments that are happening during chronic inflammation could lead to cancer. And today I'm going to be focusing on the parts that I am playing in this collaboration, which is looking at the impact of this remodeling on the behavior of intestinal stem cells. To determine changes in intestinal stem cell dynamics, our lab and others have been using a technique that is called lineage tracing. And this consists in marking a stem cell with a specific color that can then be passed on to their progeny. And this way we can follow the expansion or the replacement of a clone in a crypt. A clone here is referring to a stem cell in its progeny. So in this case, when the clone completely fills up a crypt, we say it has been monoclonally converted. But we can also follow the expansion of clones outside of the crypt through a process called fission, by which one crypt can divide into two and then more. So using this method, it was first shown that in homeostasis or in normal condition, each stem cell has the same chance of replacing another stem cell in the crypt. So as a result, wild type or normal clones are not advantaged over other wild type clones in terms of monoclonal conversion or in terms of clonal expansion. And this is called the neutral drift model. However, using the same method, it was then shown that clones that bear pro-oncogenic mutations, such as KRAS or APC, are actually advantaged in those same processes. And here you have the example of a KRAS activating mutation, KRAS G12D, that's very prevalent in colorectal cancer. And as you can see on this image, if a clone is mutated for KRAS, that's the bottom panel, the dye will fill up the crypt faster. So the crypt is monoclonally converted faster than when we are looking at a wild type clone. And this is also represented or reflected in this graph, showing the average clone size for a KRAS mutated clone in red compared to a wild type one in gray, where the clone size is on average larger for KRAS mutated clones. And so this quantification revealed that the probability of replacement of a KRAS clone by a wild type clone is only 22%, while the probability for a KRAS clone to replace a wild type one is 78%. This is very relevant because it means that clones bearing pro-oncogenic mutations are able to expand and to get fixed in the tissue faster, and therefore they can create fields in a tissue where the risk of tumorigenesis is increased. That's the definition of field cancerization. Now, in inflammatory bowel disease, the consensus, and this is data taken mostly from these 3 papers published in Nature in 2020, the consensus is that the mutational burden in the colonic epithelium of IBD patients is increased 2.4 to 3 times compared to healthy patients' epithelium. On this first graph, you can see the number of mutations found in patients compared to their age. And in blue, you have control patients, and in red, ulcerative colitis patients, where you can see the mutational burden is increased. And this rate was found to be even higher in dysplastic tissues, which is considered to be a precancerous lesion in IBD. So the reason why I became very interested in these articles is because the increased mutational burden that they saw was also associated with extensive clonal expansion, sometimes even spanning centimeters of a patient colon. And you can see this in the picture here. So this is the clonal landscape of the colon of a patient with no history of ulcerative colitis, where there is a very healthy mix of different clonal population. And this is contrasted with the clonal landscape of the colon of a patient 14 years after the onset of ulcerative colitis, where one clonal population here in green has overtaken this part of the colon. And in these papers, they also looked at the prevalence of different mutations in IBD tissue compared to colitis-associated cancer tissue. And here you can see the prevalence of these mutations in ulcerative colitis in black compared to colitis-associated cancer in pink. And what I want you to take out of this is that most mutations that were selected for in ulcerative colitis were part of the IL-17 signaling pathway, and they were not prevalent then in colitis-associated cancer. So these expansions probably are helping the tissue to repair after damage. But there are also some pro-oncogenic mutations that we see quite prevalent in the ulcerative colitis epithelium and in colitis-associated cancer, such as KRAS. So the question that I am asking here is, could the context of chronic inflammation not only favor the selection of mutations that could be beneficial for tissue repair, but also could they favor the expansion of clones bearing pro-oncogenic mutations, leading to field cancerization and really explaining why these patients have a higher risk of developing colitis-associated cancer. So what I want to talk to you about today and show you is, first of all, a description of the tissue landscape in the mouse model that we are using to study colitis in vivo. Then I want to show you data on our quantification of clonal expansion in this model and how we are associating it with markers of pathology. And finally, I want to show you some data on how chronic inflammation pathology can affect tumorigenesis. And this is done in models of colitis-associated cancer. So to model chronic colonic inflammation, we have been using a mouse model with a constitutive knockout of mucin-2. That's a very important component of the mucous barrier, both in mice and in human. And you can see we achieved a complete knockout of mucin-2 in Muc2 homozygous mice. And this leads to an impairment of the production of staining. And when the mucous barrier is impaired, what happens is that bacteria and microbes can reach the epithelial layer, and this triggers chronic inflammation in the colon. So here, the Muc2 homozygous mice have developed clinical signs such as oscillating weight and diarrhea. And the most common human endpoint for these mice is the development of an anal prolapse. And here I just showed you a hematoxylin and eosin stain of this tissue. So this is colon tissue that we Swiss roll, so we can see the full length. And you can see, I hope, that the Muc2 heterozygous mice look normal. So we are using them as wild-type controls. Now, the signs of pathology that we see in these chronically inflamed mice are hyperproliferation or hyperplasia of the epithelial cell layer. This is actually sometimes even macroscopically visible, so we can see it without even the need of a microscope. We also see immune infiltrates, as well as abscesses and different grade of dysplasia. And here, this is a very detailed spatial analysis that we've done looking at these different inflammatory hallmarks, where we were able to define two regions where the inflammatory burden was higher. So one in the proximal colon and one in the distal colon. And this is contrasted with the middle part of the colon, which actually retains a normal epithelial structure, and led us to use it as a matched normal control, so an internal control. In an effort to characterize extracellular matrix remodeling in this model of colitis, we decellularized the colons of 5 Muc2 homozygous mice that were taken at endpoints, and their age-matched controls, so Muc2 heterozygous mice. And this decellularization process allows to remove different cellular fractions at each step. So we start with removing cytosolic proteins, then nucleoproteins, membrane, and finally cytoskeletal proteins. And we end up with an ECM-enriched fraction. And this is what we sent to our collaborators for mass spectrometry analysis. And here we had taken samples from 2 regions along the colon, the inflamed proximal region I mentioned just earlier, and the middle matched normal, and then similar regions in our wild-type controls. And one of the first observation that we made was that actually the location of where the samples were taken was what accounted for most changes in protein expression. So there were 295 proteins that were significantly differentially expressed in the proximal compared to the normal colon of wild-type mice. And what we also saw is that there were only 107 proteins differentially expressed in the proximal versus middle colon in our chronically inflamed mice. So this should suggest that there is a loss of these region-based differences in the expression of extracellular matrix proteins in inflammation. This potentially suggests that what we thought as a matched normal middle control was actually more inflamed, or maybe presenting a pre-symptomatic case of inflammation. So to determine the relevance of our extracellular matrix modeling that we see in the mouse model of colitis, we compared the expression of a signature set of ECM targets that were found in different models of chronic inflammation-associated cancers, so human patients. And these are the ones that are studied in the wider cancer team. So I removed the names of the target here for confidentiality, but I hope that you can see that the changes in expression matched closely between our model, which is the last column here, and the different colitis-associated cancer samples. So of course, there are confounding factors here, such as looking at different organisms, but this is actually something that we want to relate to see if our mouse model could actually match what we see happening in human. These are also different organs, so obviously there's going to be a bit of struggle with matching the proteins, and potentially different pathological grades, because we are here looking at cancer in the human versus chronic inflammation in the mouse. But overall, the remodeling of the extracellular matrix that we saw in our mouse model of colitis seem to be relevant to what we can observe in chronic inflammation-associated cancers in patients, which is important in the validation of this model. And we were also able to validate the expression of some of these proteins. This is here the example of MMP9, which was validated by immunofluorescence for its increase in chronically inflamed tissue. So after having described this model, we wanted to be able to quantify clonal expansion in chronic inflammation. So what we did is breeding a Villain-3 confetti model onto our Muc2 knockout model. This consists in a multicolor reporter, so the confetti, which is inducible by Cre recombination through an intraperitoneal injection of tamoxifen. That's directly injected in the body cavity. And this recombination event is only going to happen in intestinal cells because we have the Villain promoter at the beginning of this construct. So practically speaking, what is going to happen is a certain percentage of cells are going to express a specific color. So here it's red, blue, and yellow. We can even have more color combinations if the confetti is homozygous. And only the long-lived cells, such as the stem cells, will be able to retain this dye and then pass it on to their daughter cells, which is how we trace their lineage or their expansion. So here, a typical protocol would be the injection of mice with 4 milligrams of tamoxifen, and then aging them until a human endpoint, which is usually the development of an anal prolapse. At which point we will collect the colon tissue and we actually optically clear it. So we make it completely transparent before we can image it under a confocal microscope. So this is the type of image that we get. And I hope here that you can see the increased number of large clone sizes in the colon of aged Muc2 homozygous mice. So this is not something that we see at early time points, but as the disease progresses and it extends, we can see more and more of these events, and they also become larger. And when we are looking at the proportion of these large clonal expansion events compared to the time that the mice have survived, first of all, we recover this idea of the clonal expansion being minimal when the mice are asymptomatic or before the inflammation starts. And then it increases very fast to reach a sweet spot, which is possibly the height of the inflammation. But you can also see that when mice survive longer than the average survival for these Muc2 homozygous mice, we can actually see less clonal expansion events. So this can mean potentially that the inflammatory burden was just lower in these mice, and this is why they survive longer, and this is why they have less clonal expansion events. Or it could also mean that clonal expansion isn't always as protective in the context of chronic inflammation. So this idea, and also the variability in the location of the expansion events that we see in chronically inflamed tissue, as you can see here, is why we decided to look for markers of pathology that we could associate with the presence of these expansion events to determine their relationship to pathology. And we did that with a characterization of the transcriptome of Muc2 homozygous mice, so bulk RNA-seq, either in asymptomatic Muc2 homozygous mice or in mice taken at endpoints, and always with age-matched controls. And this was done in bulk samples of colon tissue, so that allowed us to keep a stromal component to the analysis and not only be looking at the epithelium. And I'm not going to go over all of the details of this analysis, but one observation that really jumped at me at the time was the strong upregulation of a fetal marker signature in the chronically inflamed mouse colon. And you can see here, this is the result of a gene set enrichment analysis for a set of fetal markers. And this shows very strong enrichment. And then this figure is just showing you examples of such targets. And this was interesting to me, because at the time I was also reading a paper in which the fetal genes actually had been shown to mark the regenerating epithelium after DSS-induced colitis. So in this case, the DSS treatment is what impairs the mucus barrier. And this is arguably a more acute type of inflammation than in our Muc2 knockout model. But regardless, this was very interesting because here the tissue regeneration was depending on extracellular matrix remodeling. So we decided to investigate the tissue expression of some of these markers, the ones that are mentioned here actually, in our Muc2 knockout chronically inflamed tissue. And this is what I'm showing here. So the fetal marker TROP2 was actually very interesting to us because it was already being used by one of our collaborators as a marker of dysplasia in chronic inflammation-induced gastric cancer. So this is a calcium signal transducer, and it's upregulated in most epithelial cancers, which is also why it's an interesting target. And ANXA1 or annexin-1 is a calcium-dependent phospholipid binding protein, is actually mostly expressed by immune cells, and it acts by inhibiting the synthesis of pro-inflammatory molecules. So here you can see TROP2 in red and ANXA1 in yellow, and I hope you can see that both of these were expressed only in regions of the tissue that look more dysplastic and inflamed. And on the quantifications here, you can see that these are only found in chronically inflamed tissue. And remarkably, for TROP2, its expression was also very specific to glands that are invading under the muscularis mucosa. And that's the uppermost layer of muscle in the colonic mucosa. And we actually quantify the number of these invasion events that we could see in our chronically inflamed mice, which was significantly increased compared to wild-type mice. And this quantification here shows you the percentage of invading fronts that were expressing TROP2 in different mice, and it's always at least 50%. So this really underlines the capacity of TROP2 to mark not only chronic inflammatory related pathology, but very advanced pathology. Now to look at the relationship between these pathology markers and clonal expansion, what I did here is use a software to draw layers first around the region expressing these markers, and then in regions going away from that area of interest, and quantifying clone sizes in these different regions. And I hope you can see here on the graph that the larger clone sizes were associated with the presence of these pathology markers. So now that we established the correlation between the presence of pathology and the clonal expansion events, what we wanted to do was study their relationship in models of colitis-associated cancer. So to do this, fairly recently, we've been using the chemical mutagen ENU. So this is actually used routinely in the lab by Philippe, who is a postdoc. And this chemical mutagen is creating small breaks or point mutations in the DNA. And in wild-type mice, these are not affected by the treatment with ENU in terms of survival. They might develop a few intestinal tumors, but usually none in the colon. However, when we give ENU to chronically inflamed, so Muc2 homozygous mice, these present with a very strongly reduced survival and also the presence of many colonic tumors. So this is really showing that chronic inflammation is able to prime the epithelium for tumorigenesis or neoplasia. And what is very interesting with this model as well is that the tumors we can stain them with beta-catenin because they express it very strongly in their nuclei. So this is very useful for us to locate the tumors, which is usually made very difficult by their characteristic being flat and also the complex mixed pathology in our model of chronic inflammation. In terms of phenotypic characterization of these tumors, we have looked at the expression of our fetal or pathology markers, ANXA1 and TROP2. And here you can see their expression in inflammation on top versus tumor on the bottom, where especially with TROP2, you can see that it's very specifically present in tumor glands. And I think this also goes back to the characteristic of these tumors. Not only are they flat, but they are very dysplastic. And this really matches the characteristic of tumors that we see in patients of colitis-associated cancer. Finally, we did do a spatial analysis of different inflammatory hallmarks and tried to correlate it with tumorigenic hallmarks, so the presence of tumors along the colon. And that showed that the tumors were forming in regions that are inflamed, so presenting with immune infiltrates, abscesses, and hyperproliferation. Their location also correlates with dysplastic regions, which is actually very fitting because we know that this is a pre-neoplastic lesion in colitis-associated cancer. And this was all very interesting because these are the same regions that we see being host to clonal expansion events. So this really consolidates the idea that there is a relationship between clonal expansion and tumorigenesis in inflammatory bowel disease. Finally, I did wanted to show you some other models of colitis-associated cancer that we have developed. And these are associating specific bronchogenic mutations. So these are the ones that are prevalent in colitis-associated cancer. And here is the example of KRAS. And we associate them to chronic inflammation. So in the case of KRAS, doesn't seem to have a massive impact on survival, although it could have some small impact in early time points. But what's important, it's that it leads to the formation of lesions that are very invasive and with a very strong expression of TROP2, the marker of dysplasia. So to conclude, I hope that in this presentation, I've showed you that clonal expansion in chronic inflammation was linked to very advanced pathology, and that we were able to show that both beneficial stem cell expansions, the ones that are necessary for tissue repair, but also detrimental ones originating from cancer drivers, mutated cells, are coexisting in the same region of the chronically inflamed tissue. Finally, the bronchogenic mutation seems to be able to benefit from this chronically inflamed environment and promote the formation of very advanced and invasive lesions. So this whole, I think, highlights the need that there is to create a dual approach to clonal expansion by trying to limit the expansion of cancer-driver-mutated cells, but not limiting the expansion of protective expansions that are necessary for tissue repair. So the way that we are moving forward now from this and continuing the work from my PhD is to try to find ways that we can quantify these events separately, and also find molecules potentially in the extracellular matrix that we can use to target both of these types of expansion separately. So this is the end of my talk, and I just wanted to acknowledge, obviously, everyone in my lab, the Winton group, very helpful for their mostly advice on my project. All of the core facilities that we have in the Institute are priceless in terms of helping us to achieve our projects. And the Storming Cancer team is also a massive help in terms of collaboration. And we are really trying to align all of the techniques that we are using to all of the types of models that we have. And this is very, I think, going to highlight the different processes that are related in different types of chronic inflammation-associated cancers. So, and thank you very much for listening to me, and I can answer any questions now.
Thank you so much. That was really interesting and really, really clearly presented. So particularly for those of us who aren't sort of cancer specialists, that was really, really well explained. So thank you so much. So does anyone have any questions? Feel free either to raise your hand and then you can unmute, or you can pop them in the chat and then I can ask them to Elisa directly. Perhaps while those questions are coming in, one of the things that I was sort of thinking on in terms of the markers that you started looking at, so the TROP2 and the ANXA1, are these types of things that you could target therapeutically? Do they have other sort of physiological roles as well?
Yes, TROP2 is actually a very interesting one in terms of cancer, obviously, and it has been targeted previously. There's actually some studies done using a MEK inhibitor, which is the pathway, the ERK pathway is what is being targeted by TROP2. And this is something that we are also using in the lab quite routinely. So it's something that I'm investigating at the moment. It's definitely possible to do.
Great. And what sort of targets, you mentioned you're now looking at things that you can target, the sort of beneficial clones versus the more damaging clones. Are you starting to get any sort of inclination of what types of genes they might be?
So the way that we want to do this is first quantify what is really the difference in terms of expansion between these two types. So we have been trying to develop some molecular tools to be able to quantify in our models the recombination that we have. So for example, I can go back maybe. Yep. So with that, we have this model with KRAS activation in the context of chronic inflammation, and we can potentially quantify the recombination of the KRAS mutation and compare it to the recombination of our confetti marker as a proxy for the normal expansions. So this is what we're trying to develop at the moment. And then in terms of targets that would potentially have a differential impact on these, there has been talk recently of maybe doing some types of more targeted single cell RNA sequencing, looking at populations that we know to be pathological, such as the TROP2 population. And that might give us insights into how these expansions develop in contrast with the expansions that do not look dysplastic, for example.
Yeah, okay, that sounds great, thank you. Does anyone have any questions for Elisa? Everyone's seeming quite quiet today. In that case, while people are continuing to think, so one question that generally we quite like to ask the speakers in TMS, and particularly those who are at early stage of the career, you're obviously doing your PhD at the moment, and no pressure to have an answer for this, but do you have any ideas of what you want to do next within your career, whether you want to stay in academia or what you're thinking about?
Something I actually think about quite a lot. Obviously, I'm in my third year at the moment, finishing my third year, so I still have a year of my PhD. So this is really the moment where I'm looking around and seeing what's possible to do. And I've never really considered going into anything else than academia before. So I really always had this path in my head that I would go on to do a postdoc. So I think at the moment that's still what I want to do. But obviously, this all depends on the quality of the work that is being produced at the end of the PhD. So this is something that I will have to potentially reconsider in a year time. So
yeah. It's always the question, isn't it? Whether it's something you want to do, but also whether the results come in to enable.
Exactly.
Yeah. So we do have a question here from Hannah. So she's asked, are you aware of any risk factors for IBD which people can avoid to reduce their risk of cancer development?
Well, most risk factors are, to my knowledge, is mostly related to age. And as we don't really know the cause of IBD, it's tricky to say because people have been trying to link the development of IBD to diet just environmental factors, but that doesn't necessarily seem to correlate well. So we know that it can worsen the pathology, but we don't know if it can actually develop it. In terms of reducing the cancer development risk for people that already have inflammatory bowel disease, I don't think we have any idea at the moment of the risk factor, because we don't actually know why these patients develop cancer at this point.
Great. That's great. Thank you. And really good question. We have another one here from Oriana. She said, I think one of the treatments for serious ulcerative colitis is bowel resection. Are these patients still at risk for cancer in their remaining bowel? That's
very interesting. It's a very, very good point. I think this is something that we might potentially be able to answer looking at different regions of the colon. So not only focusing on the regions with the active pathology and regions that are potentially the ones where the tumors are going to form, but also looking at the more normal, match normal and adjacent types of tissue that potentially could be the one remaining after a resection.
That's great. Okay, did anyone have any other questions at all for Elisa? Okay, if not, then we can close it here. And thank you so much for a really interesting talk and great answers to those questions as well. So thank you for that.
Thank you so much for the invitation.
No problem. And we will see you all next week. Thank you very much.
Bye.